Good afternoon and welcome to our webinar today on how water systems can go wrong and what to do about it. Before we start, I have some housekeeping bits and pieces to go through with you. If you have any technical issues, please send a short message in the questions box, which should be on the right-hand side of your screen, and we will try and help where possible. During the webinar, we would like to invite you to ask as many questions as you would like using the questions box also on the right-hand side. And at the end of the webinar, time permitting, we'll try to answer as many of these as possible. Any that we don't get round to, we will answer offline and share with you, along with a copy of the slides from today. So, good afternoon. My name's Annette Russell, and I am the Sterile and Non-Sterile Microbiology Lead at RSSL. I've worked in the commercial team at RSSL here for over 6 years, and my role is to support our clients with a variety of microbiological testing, from routine microbial limits testing all the way through to more complex environmental monitoring assessments and the associated testing. So, for those of you who don't know who RSSL are, we are a company based in the Reading area, which is about 40 miles out of London. And we have over 250 scientists working and supporting clients in 12 different technical areas, of which the microbiology area is just one of them. And we support clients globally around the world. We even have clients as far away as Singapore and Australia. Our strengths are being that we are a relatively compact CRO based in the Reading area, so we have a cross-site agility to manage multi-disciplined complex projects. We also have the backing of our multi-billion-dollar parent company Mondelez, which enables us to have access to funding to support expansion and development. Our diversity means that we can support a wide range of biological, microbiological, chemical and physical analytical services across all phases of your product development, from clinical all the way through to commercial release. We are exceedingly well placed to support all of the new Annex 1 regulations, and hence why we're putting out these webinars with the help of Tim. And we are lucky to have a knowledgeable team who can both advise and support you if you have any issues. But that's enough of me and RSSL. I'd like to introduce you now to our guest speaker, Dr. Tim Sandle. I'm sure many of you have heard of Tim and have subscribed to his publications. Tim has over 25 years' experience of microbiological research and biopharmaceutical processing. He is a member of several editorial boards and has written over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK, but he also finds time, and I'm not sure how, to be a visiting tutor at both the University of Manchester and UCL. Over to you, Tim.
Okay, thank you very much. Okay, has my screen come through okay?
Yes.
Okay, brilliant. Well, thank you very much and welcome everybody to this webinar, which is going to take an overview of pharmaceutical water systems, particularly the risks that arise from microbial contamination, and we'll have a look at some of the best remediation actions that we can consider. So we'll begin with looking at microbial contamination in water, some of the organisms that we might find and some of the risks that they present. We'll then look into what lessons we can learn from design and qualification. In particular, maintaining those aspects is very important. We'll look at the key points for validation of water systems, we will consider effective controls and practices, and then we'll look at the particular risk posed by biofilms and some of the remediation actions, and then just finish with how we can assess whether our water systems are behaving correctly through trend analysis. So most water systems will contain a level of microorganisms. So I suppose arguably a hot WFI system is probably the only aspect of a water system that's going to be free of organisms at any kind of level. And what is of concern is with the numbers of microorganisms that we might be detecting, as well as the frequency of the incidences of those organisms. And for particular concerns with non-sterile pharmaceutical processing, we'll also have concerns about the presence of organisms that can cause a high level of patient harm, so what we may well call objectionable organisms. And there's been a lot of focus recently on Burkholderia cepacia, or the Burkholderia cepacia complex of these 20 related organisms, as well as other pathogens like Pseudomonas aeruginosa. And water not only presents a direct contamination, so if we are using water and that water is not of the appropriate standard, then that is something we can use within cleaning and within product formulation. We also need to be mindful of residual water and leaking water and the presence of water within the clean rooms as well. But contaminated water also presents concerns because it acts as a growth source for certain microorganisms, allowing them to survive under these low nutrient conditions and potentially to multiply. And it's also a vector for spreading contaminations, as might occur through aerosol generation. So we have this dual problem that a number of microorganisms will grow in water, and water spreads microorganisms relatively easily. And we have a particular concern with gram-negative bacteria. So gram-positive bacteria are rarely, if ever, recovered from water systems. So the act of water sampling can sometimes detect gram-positives, but that's normally things associated with sampling technique or around the sample outlet. But within a water system, it's non-fermenting gram-negative bacteria that are going to be the most common. And these are the organisms that can survive and grow and are often adept at existing in nutritive depleted conditions where there is a carbon source, but it's a very weak carbon source. And there's also going to be organisms present in two states as well. So we have what we refer to as the planktonic state, which is organisms that will be freely floating in water, and those that are in the sessile state, which is attached to a surface. And it's that attachment to the surface that is the basis of the biofilm formation, because those organisms can then begin to secrete a sticky polymer-like substance and start to gather other organisms and develop a community. Now, the majority of true contamination in a water system will see most organisms attached to surfaces and a lower number that are free-floating. And that's because there's a preference for organisms to, if they're going to remain within the water system, to attach to a surface and colonize that surface. So, as I've discussed, the microbiome of a water system, or certainly pharmaceutically treated water, is going to, where it is contaminated, consists mainly of gram-negative organisms. These tend to be related to the Pseudomonad or they were variants of pseudomonads in the past, and they're the gram negatives that we'd classify as non-fermenters. So as a very crude kind of differentiation, we can group gram negatives into fermenters, which are the types of things that we find within our body, so the enteric organisms such as Escherichia coli, and the non-fermenters, which are the pseudomonad type organisms, which are more common to soil and to water systems. And our fundamental concern is avoiding contamination. Where we have contamination, it's ensuring that the water is moving sufficiently fast to prevent colonization, to have conditions that are not conducive to growth. So that's trying to eliminate or bring carbon sources down to the lowest possible level, to prevent biofilms and then avoid biofilms slowly releasing bacteria, and also producing organisms that can then break down to produce bacterial endotoxin. So I mentioned with the objectionables that there is a requirement within Pharmacopoeia to look at the particular products that we are manufacturing for non-sterile pharmaceuticals, to look at the patient population, to look at the likely contaminants and sources of those contaminants, and to define a list of objectionables and a way of then assessing those objectionable organisms. And the objectionable that's received, or the group of objectionable organisms that's received the most attention, is the Burkholderia cepacia complex. So this is a complex of around 20 interrelated organisms, or biovars, and this organism poses a particular risk of contaminating water-based drugs. And for these reasons, particularly in the US, the FDA has put out a lot of detail on the screening for and avoidance of this organism, particularly in the risk it poses to inhalation products, and that risk that then arises to immunocompromised patients. So there is particular concerns on manufacturers in establishing procedures in terms of good sanitary design, equipment cleaning, and so on, to prevent this cross-contamination. And there is a USP chapter which describes a culture medium for screening of Burkholderia cepacia. That's USP chapter 60. Okay, so that's some sort of general background and trying to set the objectives of what we're trying to achieve with the water system control. In this next part, we're going to have a brief look at water system design and qualification. So on the screen there is an imaginary water system, but one that's fairly representative of a standard water system designed to produce pharmaceutical grade water, be that purified water or water for injection. So there are a number of key aspects of that process that we need to look at. So there'll be a break tank, an activated carbon bed, a water softening column, deionization columns and devices. And each one of those presents a risk of contamination if it is not properly controlled. And then depending on the purification step, we'd either go to reverse osmosis or reverse osmosis followed by distillation. And we'll talk about, in particular, the different ways of generating water for injection later. And then the water needs to be distributed and stored. And it's very important in that design process to have sufficient controls to avoid contamination. And some of these controls are factors like having smooth internal surfaces, particularly inside water holding tanks and in pipework. And this is necessary because microorganisms will adhere less well to smooth surfaces, and they would adhere better to rough surfaces. So in particular, with stainless steel, then we can assess the surface roughness value and ensure that we're specifying material of sufficient smoothness, which is expressed by values like RA, where we can measure the degree of roughness in micrograms, and specify stainless steel generally of a value of less than 0.8 RA. But we also need to be concerned with other design factors. So we need to be concerned about anything that can cause the water to slow down or can cause the opportunity for microorganisms, if they're present, to adhere to the surfaces. So pipe joints and welds are of concern, and these can disrupt smoothness. But we also need to have the continuous movement of water through pipes. So if we have rapid turbulent flow of water, then we again will avoid areas of stagnant water and avoid that opportunity again for biofilm formation. And although we might have sufficient turbulent flow, which we would express through the Reynolds number, and we'd be seeking 2,000 to 3,000 Reynolds turbulency and velocity, where we have dead legs or water branches that come off from the circulating main, these can sometimes cause the speed and the turbulence of the water to slow down, and this can lead to contamination. And we also want to ensure that the shear forces are sufficiently high, because again, under conditions where shear forces are high, then it makes microbial attachment far more difficult. We also need to be careful with valves, and in particular point of use valves and tap valves, that these are of a sanitary design, and also if they're not in frequent or regular use, then that they are subjected to a degree of flushing. Now with water storage tanks, then these need to be suitable for their use in terms of their capacity, rate of use and frequency of flushing, and the sanitisation method to which they're subjected to. And it's been shown that regular water turnover is a key aspect, again, in preventing contamination, whereas slow turnover leads to the greater potential for contamination. So if we do have slowdowns in production or periods of outage, then it's still really important to make sure that the water system is turning over at a relatively high rate. It's also important to prevent microbial contamination from the outside air, that the tanks are vented and the vents have hydrophobic air filters on them. And Annex 1 calls out the importance of checking these particular types of filters and vents, and these should be subject to periodic integrity testing. Annex 1 doesn't call out any particular frequency, but in my opinion, something should be looking at every 6 months. Further to the design, the ring mains, the main circulating aspect of the water system, should have a drop from the point of origin to the point of return, and that helps to ensure that the system is easily drainable. And we need to have regular inspections and continuous monitoring of pressure, and these go towards the avoidance of leakage as well as human observation. And water leaks are a concern because they cause the bridging of water from the external environment through which bacteria can enter into the system. And simply saying that the system stayed under pressure is not in itself sufficient, because a lot of organisms are quite adept at crossing the barrier. So it's not the same way that we might look at a strong pressure from one clean room to another. It is very difficult for air to cross, but with water, it's far easier to bridge and to gravitate contamination over. So there's important concerns with those regular inspection points. So we mentioned about the good distribution of water and having the smoothness of the tanks and the pipework, and the continuous movement of water, and the importance of avoiding pockets of stagnant water or dead legs, and the importance of maintaining valves and of avoiding leakage. So they're all key reminder points that are put on there. In terms of user outlets or point of use, taps or tubing. So the idea here is, particularly within the manufacturing area, is to transfer water from the circulating water loop to its point of use via some form of transfer device, which is typically tubing. So it's important that the tubing is formed from non-toxic material, and polyvinyl chloride or polypropylene tend to be common choices. It's important that the tubing is subject to decontamination, so it's ideally autoclaved before use, and is only used for periods of, say, up to 24 hours. It's also important when tubing is not in use that it's hung up, so it free drains and not left attached to the outlet. We also need to take concern and care when using the outlet, particularly avoiding splashback from sinks or other activities that can lead to recontamination of aerosols. It's also good practice just before using and committing water for manufacturing use to flush it, but this should be for a defined time or a given volume of water or some combination of the two. And it's very important that the quality control sampling is identical to the same amount of time or flush volume that the manufacturing staff would be subjecting that water to. Otherwise, there's a risk that the water is unrepresentative, and that can lead to missing contamination. So I just have on the slide there just an example of poor design. So this is a water outlet with a clear two examples of where dead legs can occur and where there's a risk of biofilm building up. So it's those sorts of poor design elements that we need to be mindful of. We can also pick up some contamination concerns from regulatory activity. So on the slide there are some extracts, some points raised from US FDA warning letters. And these pick up some of the points that we previously mentioned, such as water systems not continuously circulating, or the monitoring not being representative, or insufficient samples being taken, or relating to the test methods or objectionable organisms, and so on. So these feed into some key design elements, and in addition to that, it's important to ensure that things like carbon beds are regularly subject to sanitization. So we're applying appropriate sanitization to the water generation plant, and then ironing out pockets within the design process that could lead to opportunities for contamination to arise. So that's the dead legs and long undrainable pipes and U-bends and so on. And it's very important, and this builds into the Annex 1 requirements for the contamination control strategy, that we are thinking carefully about the risks and developing a rationale to consider the various elements. So we need to undertake a quality risk assessment and ask ourselves, what are the risks? Consider whether they are or not adequately controlled. To then consider what the sanitisation procedure is and the frequency of sanitisation and whether that is itself adequate to address the risks. And that includes both water generation and water distribution. And then we also need within the rationale to ensure that our microbiological monitoring is also suitable and representative and sufficient to give us good detection. So we need to ask questions about when to sample. So if it's a 24-hour facility, then the sampling needs to be reflective of different times of use. Taking a reasonable sample size, and that is the pharmaceutical minimum of 100 or 200 ml, ensuring that we're using a proven test method and that we are satisfied that our method can detect contamination, particularly of organisms that might be nutritively depleted or undergoing some form of stress response, and that then our identification strategy is suitable for understanding what the water microbiota is, and in particular where we're interested in screening for objectionable organisms. It is also important that we have subjected the water system to a good validation, and that we are regularly assessing, as part of the life cycle approach for validation, the status of the system and calling in a revalidation should the data suggest there is a problem. And we take a water system through a number of requisites. So we need to make sure that we're using quality by design and getting the design elements in at the beginning. We're going through the validation documentation hierarchy, and we're completing each stage appropriately. And with water system qualification, we would break that down into different phases, and we should be designing a sampling regime that starts with the supplied mains water and samples after each step of the treatment process, includes each point of use, and includes water that is the point of being supplied and the water that is the point of return. And during the initial phase of any water qualification, then sampling needs to be daily for both chemical and microbiological monitoring. And this will then allow operating and testing SOPs to be generated, and we will be doing that level of testing over a 2-week period. That would then move on to the second phase, which would have a typical duration of 2 to 4 weeks. And here the aim is to demonstrate we can achieve consistent operation within our established ranges, be that chemical or microbial limits, and to demonstrate the consistent production and delivery of water of the required quality. Now, the sampling scheme would still be typically as of phase 1, and we'll still be looking at daily monitoring. For the third phase of water qualification, then this would extend to 1 year, so that we can capture seasonality, as in variations in the quality of supply water and the ability of the water generation plant to handle varying levels of microorganisms, but also gives the opportunity to test out the standard maintenance schedule as well, and to show that we can effectively treat water of the required quality over an extended period of time. And we would need to use the information from the previous phases to determine the sample locations and tests. And a typical frequency would be weekly at this point. There's also of importance that following the completion of a qualification of a water system, that any changes are managed as per the change control system, and also that all laboratory data is being trended, and that there are systems in place to investigate variations in trends, so that we can answer questions relating to maintenance, seasonal source water variability, routine maintenance events, the effectiveness of sanitization, over things like a rolling 1 year period, and become experts in understanding the water system and what it is and isn't capable of. In terms of the sort of post-qualification frequencies, there's going to be variations depending upon the particular pharmaceutical processes and nature of the operations. And there are lots of variations in industry from daily through to monthly. But typically, a purified water system would be sampled at least monthly, and a water-free injection system would be sampled at least weekly, with the proviso that the supply and return is assessed each day of operation, and there's a number of user outlets that are looked at each day. And Annex 1 in particular signals to us the importance of assessing the water system regularly during different points of use. Okay, so we're now going to move on to look at some of the controls and practices that we need to keep in mind. So here on this slide, there's some examples of bad practices. So on the left-hand side, there's water tubing that's attached, so it's not in use, but it's not been disassembled and allowed to free drain. And on the other side is water tubing that has been disattached, but it's not free draining, and part of the outlet is in contact with a rather dirty-looking dishcloth. So some of the things that are not particularly good practices. And we can get a feel for what are the good and bad practices by undertaking regular audits of our water system. So this is something that quality assurance, microbiology, engineering and production can do in harmony. And here it can be useful to use drawings. So look for inconsistencies with drawings of water systems, and then go around and actually look at what is present within the facility. And that can be quite good at, are things matching what we think they are? To look at point of use, particularly the things I mentioned about tubing, but also making sure we've got sanitary sampling valves and we don't have valves where there's water sitting in them. To look at some of the ergonomic issues, so can those who are required to use the water or sample the water actually get to the water. Consider how water is drawn. So if there's a requirement to flush the water, can it actually be flushed? Is there a nearby drain, or are buckets being used? And if so, are those buckets in the water? Are the buckets sitting there with stagnant water as well? Also look at certain operational requirements. So heat exchangers is an important one. So if a heat exchanger fails vacuum or develops a hole and starts to leak, that's an easier way of getting contamination into the water system. to look at things like flow controllers and volume totalizers, because they're very, very difficult to keep free of water, and again can often be sources of stagnant water and contamination. And also to review things like the sampling procedure and how's that done and whether that is itself representative. It's also a factor that certain aspects of the water system operation can be sources of contamination if they're not sufficiently controlled. So it's important to extend audit to look at potential purification problems and to ask questions of different aspects of the water system. So where we have water softeners, we need to ask what the regeneration frequency is, what the quality of the sanitizer, say it's chlorine that's used, and what the bed replacement frequency is. With carbon beds, we also need to ask what the backwashing frequency is, what the sanitization frequency is, and that's important because a carbon bed can develop into a very large colonizable niche for bacteria. Where we're doing things like bisulfite injection, we need to ask about how that works and what the microbial quality of that is. With reverse osmosis, we need to ask about the cleaning and sanitization and how often membranes are replaced and what the rationale for that is, because one of the big weaknesses with reverse osmosis is the build-up of biofilm on the dirty side of RO membranes, and that becomes an opportunity to get endotoxin released into a system. With deionization beds, then again, we should be asking questions about the regeneration and replacement frequency, the suitability of the resin. With EDI or CEDI units, we should pay particular attention to leaks. And with vent filters, as I mentioned earlier, asking questions about how often they're dried and what their integrity testing is. So there's a number of things to really delve down in to try and build the suitability of controls around the water system. But the overall, probably most important control mechanism is around the overall sanitization of the water system. And it's important to have an understanding of what that sanitization approach is and the degree to which that runs periodically. So if we're using a chemical like ozone for a cold system, then we need to understand what the frequency of that is. And if we're running a hot circulating water system, then if we ever need to go higher, what the frequency or trigger for that is to take that into sort of more superheat territory. And with the chemical sanitization, be that ozone or chlorine or some other variant, then it's really important to have understood what the concentration is, what the operational temperature is, because generally the higher the temperature, the greater the disinfectant efficacy is going to be, and how long the contact time is. And we also need to get our drawings out again and to evaluate whether any unsanitized areas are ever bypassed, and to make sure that we are able to sanitize every aspect of the water system. And also have a rationale, particularly for microbial contamination, is to say, okay, if we get so many hits or so many counts of a certain magnitude, then at what point do we instigate sanitization, and to make sure that all repairs and maintenance activities, any break into the system, any change of valves, are always followed by a resanitization exercise. Okay, so now we're going to have a look at biofilms a bit more specifically. So Biofilms develop when microorganisms adhere to a surface, and there is a period of time over which this happens. So biofilms not formed instantly, and even during the very early phases of biofilm formation, the organisms are relatively easy to remove. It's over time that they become harder and harder to remove. And that's because once these colonies start to form, then a number of the colonizing bacteria can produce, in essence, slime, or what's referred to as extracellular polymers, that facilitate the adhesion, which attract more organisms to this developing mass, and also provide a structural matrix that holds the mass together and provides the opportunity to strengthen the sort of combination of attractive and repulsive physiochemical interactions that bind the bacteria to different surfaces. And the resultant biofilm also becomes increasingly resistant to sanitization methods the longer it exists and the greater the density that gets afforded to the biofilm. And although the objective of the biofilm is to continuously building this large mass of bacteria, it will also continuously release bacteria as well, which can be as free floating cells or parts of the biofilm itself sloughing off. And this is why sometimes microbial contamination happens and then it appears to disappear, or sanitizations run and everyone relaxes and thinks the problem's gone away, and then it appears to reappear again. And that's because of this odd eluting effect of the biofilm, and that pattern of release doesn't follow normal distribution in any shape or form. So it becomes hard sometimes to predict. So with the biofilm formation, as I mentioned, it starts off as a relatively slow process and then becomes faster and then starts to layer onto layer. But it develops initially because bacterial cells, once attached, start to secrete polysaccharides. It then forms a thicker slime thing, which is referred to as the glycolax. And this also enables each bacterium to encapsulate itself and affords a degree of prevention. So during those early phases, during that phase 1 and 2, it is easier to treat a water system and get rid of a biofilm. The more mature the biofilm becomes, the harder it becomes to get rid of it. So we have also other factors at play. So the type of microorganism involved is quite key. Some organisms are more prone to form biofilms than others. Unfortunately, a lot of pseudomonads are keen to form biofilms, particularly cold water organisms like Pseudomonas fluorescens and Ralstonia pickettii. The size of the bacterial population and also the growth phase that organisms are particularly in. And gram-negatives, as I said, form biofilms more easily, and in part that's due to them often having appendages. They have little spiky protrusions around the bacterial cell called fimbriae, which allow them to attach to surfaces more easily. And also the particular charge of the organisms, often relative to the surface, is another attraction force. So we also have organisms which are used to lower nutrient environments and which require lower levels of assimilable carbon, which are also quite good at forming biofilms. And as I said, this complex this community that arises is very resistant to different forms of treatment, and there's a range of different activities which I put on the slide there, which afford the biofilm protection either from the physical forces of the water or from the different sanitization agents that arise. So biofilm remediation then becomes an issue when you suspect you have a biofilm, then becomes a problem. And it's a bit odd. There's a paradox that many of the stages of water purification create conditions that create certain strongly adapted and survivable bacteria that are then those most prone to form the biofilm. So the purification pathway does sometimes create the organisms, these complex communities that we don't really want. So anyway, how to control a biofilm? Well, there's things we can do, looping back to some of the things we've spoken about. So that with the design process, so correct materials of construction, so particularly the smoother stainless steel, the better, and stainless steel generally better than plastics. Plastics are slightly more prone to form, to allow biofilms to develop. Having the right flow rates through the Reynolds number and ensuring turbulent flow, either regular chemical dosing or maintaining a water system at a sufficiently high temperature. But a good system by itself won't prevent biofilm formation, but it will help to control it. And unfortunately, once a biofilm is there, then simply running the system at what it's required to be isn't sufficient. So a dip in performance can allow biofilm to develop. Simply ramping back up doesn't always or very often deal with the biofilm. So we've got the hygienic design and the free draining and the drain controls, and all of these factors, all these good practice things, they all contribute very little to the control of biofilms, but if they're done wrongly, they can create biofilms. So we just need to be mindful of doing things that could lead to contamination, so suck back or cutting into systems or having leaks or temperature drops or outages and not circulating the water are all things that we can do to make things go wrong that could lead to ingress of contamination and hence biofilms. But if we have a biofilm, then we have two options. One is heat and the other one is chemical, and the chemical choices are ozone and chlorine. In general, heat is vastly superior. So being able to take a water system up to a high temperature is the most effective sanitization method by far. And this then creates a problem with cold water systems. And this is why I'm not in favor of cold WFI systems. I always think you should have the option to superheat a WFI system and ideally keep a WFI system circulating at least 65 degrees Celsius. For cold systems, and this could well be a purified water system, then there is the choices of chlorine or ozone. Generally, literature suggests that ozone is superior, and keeping ozonation at 10 to 20 parts per billion with all the other factors at play can help to stop biofilms from forming, but it's very difficult to apply chemical control to eliminate a biofilm. So it's a combination of heat and system design, velocity, and so on. So the best way to tackle a biofilm is ideally with temperature and steaming if possible, or running water at 85 degrees if possible. If none of that is available, then chemical treatment is there, but often in the most serious cases, then we're left with physical removal, and you can't 100% scrub a system, so you end up having to unfortunately disassemble parts of the water system. So, and that's a situation that nobody really wants to be in. So, in assessing the biofilm risks, then heat is the most effective, chemical second, and that's partly because of the maintaining the continuity of the chemical, and partly because of things like crevices and so on. But even with hot water systems, we need to be mindful that even with hot WFI systems, then often water cools in subloops and cools as it moves down to user points. So, but cool systems in general are always going to be at greater risk. Okay, so just moving on to the last part of the presentation. I did mention trending at the beginning. It is really important to trend data over at least a year, and then to superimpose movements and rises in counts with particular events, so we can develop a greater understanding of the water system, and we can divide those into a combination of special causes and common causes. So common causes might relate to poor design of the system, the way we're managing the system, the frequency of sanitisation, that sort of thing. Specific causes can relate to maintenance work or local user outlet problems, or problems with drains or aspects of the system that are not receiving the sanitising agent as required. Okay, so thank you very much. We're going to move on to Q&A, but we've looked at overall microbial contamination, design qualification, validation, user controls, biofilms, and a little bit about trending. So I'll hand you back to Annette.
Thank you, Tim. Just while we swap the screens over, I've had lots and lots of questions come in, so I'm not going to be able to get through them all, but if we can just start with some simple ones. Had a question from Ernesto, and he said, on systems that rely on heat sanitisation, should a chemical sanitisation be performed as well?
Of the generation plant, and the best way is to use chemical sanitisation, so more likely chemicals like chlorine and so on. But for a hot water system, then that should be self-sanitising, with the water circulating at least 65 degrees or higher, and it's only a breach of the system. But even with a breach of the system, then taking that water temperature up or flushing through the system should get rid of the contamination, so it wouldn't be normal.
Okay, and I had a question from Victoria, and she said, how long can a water system be down prior to sanitising? Her water specifically is used for API manufacturing cleaning only.
It depends about the robustness of the water system, and what the possibility of contamination ingress into that water system is. So it's always going to vary, but the general rule of thumb is that the water system should ideally be continuously turning over and being drawn upon. Different companies will have different protocols on how often the time delay is before sanitisation, but I would start to get worried after sort of 24 to 48 hours. I'd always sanitise after that point, but there might be needs to do that more frequently.
Okay, and I think this is probably the last question. Is there any correlation between TOC and microbial counts?
To a degree. So all microorganisms need a carbon source. The thing with TOC is what matters to the microorganism is the amount of free carbon or carbon that they're able to assimilate. So it tends to be for like every part of TOC, only about 1 to 10% of that carbon can actually be utilized by microorganisms. So it isn't always a clear ratio, and it partly depends on the sensitivity of the tests, because it can look like you haven't really got much TOC, but there's still normally something sufficient for a microorganism to use.
Okay, that's brilliant. Well, that's all we've got time for, I'm afraid, today. So thank you to everybody for attending our webinar. I hope you found it useful. Any questions that I didn't get around to asking Tim during the Q&A session, I'll forward on to him and we'll circulate the answers with the slides. If you wish to catch up on any of our previous webinars or catch Tim's blog or regulatory update, please go to our website or keep an eye out on our social media posts. This is our penultimate webinar. Our last webinar in the series is on the 21st of June, where Tim will discuss design and qualification of isolators. As always, spaces are limited, so don't forget to register. And we also have some upcoming training courses that might be of interest on the screen in front of you now, including our annual QP conference, which includes some great presentations, but is also a really good way to network with some of the leading industry experts. So thank you for attending. And as I say, my name's Annette Russell. If you have any questions, please don't hesitate to contact me or reach out to me. Have a good afternoon.
Thank you.
Thanks.